Today, I would like to talk about
Good morning, everyone. Myself, Rijuta Ganesh Sartali. I am working in the Research Institute.
A very good morning to all of you. I welcome you all again on the third and the final day of 10th IBA conference being held in National Kaohsiung University of Science and Technology, Taiwan. So we have entered into the 8th session, which is the final session of the day 4 of the conference, and we'll be starting with the plenary lecture by Professor Yong Non Park from South Korea. He is present with us right now online, and he has also sent us the pre-recorded file. Professor Park, if you wish to present online, that would also be okay for us.
No. I want to prefer to present using recorded file.
Okay, Professor, no issues. So we'll just play the pre-recorded file, and his topic of presentation is microalga gasification over nickel-lodal perovskites for enhanced biohydrogen generation. So we'll just play his pre-recorded file, and after the presentation, if anyone has questions, he is there with us. He can ask the query. He can answer the queries. Thank you so much.
Today, I would like to talk about the microalgae gasification, especially using nickel-loaded perovskites for enhanced biohydrogen production. The world is suffering with energy needs due to the increase in energy demand and the depletion of fossil fuels. The South Korea's energy consumption is also increasing significantly with every day. Also, the excessive use of fossil fuels leave environmental footprints on the environment in the form of greenhouse gases. Hence, there is a strict need to move towards the sustainable sources. Microalgae is a third generation fuel and has a huge potential for energy generation, which is neglected in the past. The generation of microalgae is over 5 kiloton annually worldwide and can be converted to biofuels via thermochemical conversion processes such as pyrolysis, torrefaction, and gasification. The use of microalgae not only saves the water bodies but also has potential to fulfill the energy needs for future. Also, its harvestation has a benefit over terrestrial biomass as it does not require arable land and absorb CO2 at the same time. This slide provides the potential of microalgae for biofuel generation using several processes such as biochemical, chemical, and thermochemical conversions. By using this different kind of the process, we can produce different kind of energy or different kind of fuel. This slide provides the potential of microalgae for biofuel generation using thermochemical conversion processes. This diagram also enforces the idea of using microalgae for biofuel generation using different conversion processes. This slide shows the overall process of gasifications. In gasifications, a high range of temperature, 700 to 900 Celsius, and different gasifying agents such as steam, air, and carbon dioxide, or their mixture, are needed to produce hydrogen-rich gas from gasifications. And generally, biomass gasification consists of several endothermic and exothermic reactions, as can be observed in these schematic figures. Microalgae gasification is a promising technology for the production of hydrogen-rich gases as a waste-into-energy platform. Providing a partial oxidative environment and also pre-treatment of microalgae are needed before gasifications. Also, employing the catalyst can increase the gasification efficiency significantly. Generally, the efficiency of microalgae gasification directly depends on the experimental condition and type and rate of catalysis. But the most crucial problems which has been found for microalgae gasification includes these four problems. Formation of tar during gasification has been reported as the main challenge during microalgae gasifications. Handling of high water content in the microalgae is another important challenge in the gasification of microalgae. The water content of the cultivated microalgae usually varies between 75 and 98% on the wet basis. High carbon dioxide proportion and carbon deposition during microalgae gasification are considered as two other challenges in microalgae gasifications. So, importantly, these problems can be addressed using catalytic gasification with novel catalyst, especially perovskite type oxide nickel catalyst, which will be explained. As already mentioned, among all challenges of gasification biomass, for example microalgae, formation of high content of tar has been known as the main problem for scaling up the gasification systems. A higher tar yield of biomass gasification reduces the gasification efficiency and causes serious challenges such as gasification reactor pipeline obstruction, increasing the operation and maintenance costs. The current strategies for tar treatment can be categorized as dry treatment and wet treatment. But these technologies are not able to fully preserve the high gasification efficiency, given tar is wasted instead of being converted to condensable gas. So, post-processing the separate tar requires additional energy, which will further decrease the overall gasification efficiency. So, therefore, it is more feasible to directly convert tar into incondensable combustible gas, such as hydrogen. We can expect three promising strategies for tar reduction during gasifications, such as improvement of gasifier design, adjusting the operating parameters, and application of efficient catalyst. Among them, application of efficient catalyst is a very essential way to avoid the need of extreme operating conditions, to catalyze reactions, and to enhance hydrogen production with the lower tar and char. For the efficient catalyst for gasifications, the catalyst should have high activity, high lifetime, and high mechanical strength, and optimal particle size and catalyst shape, and show low coke formation. For the efficient production of hydrogen, the metal-loaded catalyst has been applied as the most common method. And for the efficient production of hydrogen, it is desirable to develop catalyst having high surface area, high pore volume, and large pore size of the support. In this regard, metal-loaded catalyst has been growing in recent years owing to the high selectivity, recyclability, and abundance on Earth. Although using noble metal-loaded catalyst is expected to have higher efficiency compared to other metal-loaded catalysts in generating hydrogen gas, the higher cost inhibits the extensive application in the gasification. So it's needed for developing trench metal-loaded catalyst, especially nickel-based catalyst, because nickel-based catalysts are cheaper and require simple regeneration and possess excellent activity. You can see the advantages of nickel-loaded catalyst. Nickel-based catalysts are effective in breaking the carbon-carbon bond, carbon-hydrogen bond, and oxygen-hydrogen bonds of oxygenated compounds of biomass gasifications. Also, metallic nickel accelerates a series of biomass gasification reactions. And metallic nickel is conductive to promote the corresponding catalytic dehydrogenation in microgasifications. However, there are some disadvantages, some limitations when you apply nickel-based catalyst in microgasifications. Firstly, nickel-loaded catalyst possess low activity for the dry reforming reactions. Secondly, catalyst deactivation due to carbon depositions on catalyst surface has remained a challenge for nickel-based catalyst. Thirdly, agglomeration of nickel particles on the surface reduces the number of metallic active sites, which reduces the catalytic activity. Fourthly, nickel-based catalyst has shortcomings of rapid deactivation by degradation of mechanical strength. So, many studies have investigated the effect of adding promoters and employing proper supports to address the above-mentioned problems of nickel-based catalyst. Among different of the supports, perovskite oxides with the general formula of AbO3 are considered one of the most promising supports for nickel catalysts due to their special characteristics. So it may be needed to use nickel as an active metal because of its cheaper price than noble metal sources. And among different supports, perovskite can be selected because it can prevent large amount of co-formation and can provide high metal dispersion, and also it can provide high mechanical stability. This slide shows the overall process of biomass gasification of a perovskite supported nickel catalyst and also its downstream applications. The developments of perovskite materials with a flexible crystal structure have received many attentions as their structures can be tailored by doping and co-doping strategies and obtain various properties accordingly. It has been demonstrated that the appropriate selection of the A and B site elements or element combination can change their catalytic activity. So properties of perovskite type oxides can be controlled by doping at the A or B sites in ABO3. The main special properties of perovskite oxide suppose are observed. Perovskite oxides indicate strong Lewis bases, hence limiting carbon deposition on the catalyst. Moreover, perovskite type oxides have the potential for ensemble size control, enhancing nickel dispersion, and hence the number of active sites. Also, these catalysts have unique redox properties and remarkable oxygen storage release capacities, which could promote the oxygen transfer and accelerate the elimination of carbon deposits. So, the employment of perovskite as support for nickel catalyst creates well-dispersed metallic particles during the reduction and limiting carbon formation, leading to superior catalytic activity and stability. The role of perovskite for improving the efficiency of nickel-based catalyst in micro gasification is as follows. Firstly, perovskites can stimulate the dry reforming reaction by supplying the structural atomic oxygen, hence converting of methane and CO2, so increased H2 selectivity. Secondly, the lattice oxygen on the perovskite support could easily oxidize the carbon and CHX fragments of the metallic particles, which accelerates the rate of reaction and elimination of displaced carbon. Thirdly, due to the presence of several metals in the perovskite structure and in its high porosity, the interaction of nickel and support can be controlled, which can also adjust the particle size of nickel. So, the dispersion can be increased, and the number of catalytic active sites also can be increased. Lastly, due to high thermal and mechanical resistance, perovskites are resistant against attrition and sintering of active metals in the nickel-based catalyst. So finally, it can increase mechanical strength. Now, I want to show some examples using nickel perovskite for the other reactions. In this slide, we applied nickel yttrium serial methane oxide perovskite catalyst in the methane reforming process. As you can see, the XLD patterns, the catalyst was well prepared, and the nickel yttrium serial ruthenium oxide catalyst is very stable and shows a rather high capability of maintaining its activity during 30 hours at 700 degrees Celsius, and has continued its activity with the least deactivations. And also, we applied nickel metal titanium oxide perovskite catalyst for the swine monosteam gasifications. As you can see, left side figures, there is no cox. So this is maybe due to the high oxygen mobility of the titanium-based catalyst, especially 10% nickel calcium titanium oxide perovskite catalyst show the maximum hydrogen selectivity. So This may be due to the well-dispersed nickel and titanium, which can enhance the water gas reactions, and calcium. So using combine these components, we can obtain the highest activity for the production of hydrogen in the swine monostin gasifications. From now on, I will introduce my recent research results using perovskite type catalyst. In this study, we prepared 3 different kind of the perovskite oxide, such as calcium zirconia oxide, calcium zirconia titanium oxide, with different ratio of zirconia to titanium ratios. And this perovskite oxide were used as supports, and nickel was impregnated on this supports. And this catalyst was prepared by gel combustion method using precursors of zirconia, calcium, and titanium. And why these compositions was selected? The answer is that zirconia can enhance thermal stability and facilitate reducibility. And calcium can boost the conversion activity, so more stability and low cork formation can be expected. And titanium can increase metal distribution and also decrease cork formation due to oxygen mobility. And so the prepared catalyst was dried and calcined finally. This slide shows the BET result. As you can see, for calcium zirconia oxide perovskite catalyst, the surface area was around 31. But if some zirconia was replaced, when some zirconia was replaced by titanium, the surface area increased up to 45. And if the nickel is uploaded on catalyst, the surface area was decreased due to the blocking pore by nickel particles. And pore size increased up to around 80 nanometers by replacement of the titanium. And this so overall, the pore size of the perovskite is around 16 to or 17 nanometers, and this pore size is larger than the alumina and zeolite. For alumina, gamma alumina, the pore size around 6 nanometers. So this large pore size allows more effective distribution of the active metal and better dispersion. In fact, the pores act as channels for accessing the inner parts of the catalyst. The ease of accessibility of gas molecules as the feed to the catalyst active size ultimately causes increased conversion as well as hydrogen production. The X-ray diffraction analysis was carried out for all prepared samples. The diffraction peaks of the synthesized perovskite are in agreement with the patterns previously reported for these samples. And lack of signals for titania and zirconia also proves no oxide impurity in the supports. This slide shows ammonia TPD for the acid amount. The density of the weak and intermediate acid size for calcium zirconia 0. Titanium 0.2 oxide, and calcium zirconia 0.6 and titanium 0.4 oxide samples can be claimed to be remarkably larger compared with that of the calcium zirconia catalyst. Therefore, the addition of titanium into these two supports reduced the acid strength of the catalyst, which along with the increase of axial mobility potential can effectively increase the stability and reduce the cork formation. Moreover, the acidic or basic nature of the support significantly affects the catalytic activity in the water shift reactions. Therefore, nickel loaded catalyst can definitely has higher selectivity to hydrogen due to the low density of its strong acidic sites. And for the oxygen vacancies, we perform O2 TPD. So this the large peak of the O2 TPD may imply that this catalyst can high oxygen mobility. So this can contribute the efficient formation in the multi-cycle redox processes. The same image corresponding to perovskite shows a spherical morphology, which can positively affect the homogeneous and uniform distribution of the active metal and prevents the sintering and agglomeration of nickel by proper interaction with the active metal, along with improving the active metal dispersion and enhance the activity and stability of catalyst. Steam catalytic gasification of microalgae was performed using this apparatus. The experimental setup consists of two furnaces. While the first furnace was movable and used for heating the feedstock reactor part, the second furnace was fixed and utilized for the catalyst upgrading. According to proximate analysis, the ash content of microalgae is more than other common biomass feedstock for gasifications. Although the high content of ash can play a catalytic role in gasification processes, however, ash content can stimulate the cork formation and also induce reverse water gas reactions. Therefore, this can necessitate the significance of applying perovskite support catalyst as their resistance against the carbon deposition. As you can see, the yield of gas increased in the case of the nickel perovskite catalyst compared to nickel gamma alumina. Interestingly, due to the high oxygen transfer potential of perovskite, there was a zero cork on the catalyst, while the cork percent of the nickel alumina was around 1.8%. This slide shows the gas composition over the applied catalyst. According to the result, the order of catalyst in the case of the hydrogen selectivity is as follows: catalyst 3, catalyst 4, catalyst 2, and catalyst 1. Indeed, better catalytic activity of perovskite supported nickel catalyst compared to nickel alumina is mainly due to their high pore volume and size and oxygen mobility potential. Also, the presence of zirconia in catalyst structure increases the thermal stability, which can lead to reduced sintering and increase the nickel dispersion. With the addition of titanium into support structure, oxygen vacancy defect can be created in the perovskite structure, which increases the oxygen mobility of the catalyst. This is the main reason of the high activity of the catalyst 3. However, by further increasing the titanium into perovskite structure, the amount of oxygen mobility will be excessively enhanced, which stimulated the reaction toward CO2 production, so decreasing the hydrogen selectivity. So summarizing my talk, microalgae has a great potential to apply as a feedstock for gasification with a target of hydrogen-rich syngas generation. Nickel-based catalysts have high catalytic efficiency in order to hydrogen generation. Employing perovskite-type oxides as support can improve the catalytic activity and stability of nickel-based catalysts in microalgae gasification. By changing the A or B element in perovskite structure, the catalytic effect of nickel-supported perovskite oxides can be adjusted and increased.
Thank you so much, Professor Park. So it was it was a pre-recorded file, but Professor Park is also here with us online. So if there are any queries from the audiences online or offline, you can ask Professor Park directly. So your question.
Good morning, Professor. It was a really wonderful talk. So I have a general query, like you are using nickel, zirconium and that kind of elements, and I'm wondering about the toxicity and reusability of the catalyst for in a long-term run.
Yeah, so I didn't show the long-term result, but when as you can see the result, the perovskite type catalyst show the high activity than nickel alumina, and so we repeated around 10 times using the same catalyst. The stability was maintained, so a small decline of the activity was observed. So I can say the perovskite type zeolite, perovskite catalyst can show the good stability for the gasification of the microalgae.
So we are moving to the next invited lecture, IL 78 by Professor Rijuta Ganesh Saratale from South Korea. The talk is pre-recorded and the title is Lignin, a valuable resource for nanoparticle synthesis, evaluation of their environment and sensing application.
Good morning, everyone. Myself, Rijuta Ganesh Sartali. I am working in the Research Institute of Integrative Life Sciences, Dongguk University, South Korea. Before I begin my research talk, I would like to thank Professor Ashok Pandey and all the organizing committee members for giving me this opportunity to present my research work in this reputed conference. So I begin with my presentation here. The topic of my title is Lignin, a valuable resource for nanoparticle synthesis, evaluation of their environment and sensing application. Before I begin, this is a view of our Dongguk University. As you can see, this is our beautiful campus, and Dongguk University is based on Buddhism, and the university name remains one of the Buddhist affiliated universities in the world. And it is also a member of International Association of Buddhist Universities. The university symbol is an elephant, which indicates the birth of Buddha, and the symbol flower is a lotus blossom, which reflects the Buddhist truth. Our university campus is also in Peckham, Gyeongju, and Los Angeles, United States. As we all know that the nanotechnology is a rapidly growing scientific field and has attracted substantial benefits with significant economic and scientific impacts ranging from aerospace engineering and nanoelectronics to environmental remediation and medical healthcare. These are the applications of nanotechnology in various sectors, mainly in the area of biomedical. They can be used as a good anti-cancer agent, antibacterial agent, antioxidant, anti-inflammatory, and for the drug delivery purpose, as well as in the anti-diabetic and bioimaging. In case of industrial, they can be used for the water purification as nano encapsulation of flavors, aromas, superplastic ceramides as industrial catalyst and nanocomposites. Whereas in case of environment, they can be used for UV protection as a fuel cell catalyst, as a pollutant scavengers, biodegradable polymer for the wastewater treatment and the contaminant sensor. And in case of agriculture, they are used as potential nanobiosensors, nanoherbicides, precision farming, improving soil texture, nutrient delivery, nanopesticide, and for the detection of pathogens. And the status of the nanomaterials or the nanoparticles in South Korea, as we can see, is widely distributed in the area of appliances, automotive, goods for children, electronics and computers, foods, health, and home and garden cross-cutting. These are the commercial already applied consumer products in Korea. For example, they are used in frying pans, refrigerator, as a water purifier for the filter, and toothpaste and other food and raw materials products. This graph shows the amount of budget, around $3.4 billion devoted for the study of the nanotechnology for the last 14 years. And in South Korea, there are certain universities like Seoul National University, Yonsei University. They are devoted toward the use of this nanotechnology in the health sector, especially for the diagnostics, that is the early diagnostic for biosensors, molecular devices, and targetable contrast agent, as well as in the drug delivery and regenerative medicines. There are certain dependent factors for the nanoformulation, mainly size, shape, material, and surface. As we all know that the size matters a lot because smaller the particle size, greater is the surface area to volume ratio, and higher is its chemical reactivity and the biological activity. Along with the size, shape also affects on nanoparticles that are likely to depend on their morphology. They could be spherical, round, pyramid, any kind of shape. And along with the size and shape, surface, because the surface, it helps in the aggregation of nanoparticles with biological material that may depend on the surface charge, mainly the surface charge or the surface coating, ligands, and the surface functional groups. Finally, the material could be hydrogel type, metal-based, dendrimer, or the polymer type, protein-based, and the carbon nanotube. So the toxicity of the nanoparticle, it depends on their chemical composition and also on the composition of any chemical adsorbed onto their surfaces. So along with these dependent factors, solubility also matters a lot because poorly soluble nanoparticles, they have shown to cause cancer and can exhibit a pronounced toxicity. And there are various different methods that have been developed for the nanoparticle synthesis, mainly the physical, chemical, and biological. Physical method involves laser ablation, ultrasonication, photoirradiation, radiolysis, spray pyrolysis, whereas chemical method involves solvothermal, co-precipitation, chemical reduction, and finally the biological method based on the type of the agent which is employed for the nanoparticle synthesis. It could be microbial, plant, algae, and the biomimetic. So green nanotechnology. Green nanotechnology is a multidisciplinary field which is serving an important technique that makes the process clean, non-hazardous, especially environmentally friendly, and in contrast with the chemical and physical methods, those are employed for the synthesis of the nanoparticles. So the green nanotechnology is what? It is nothing but the process that involve the use of do not involve the use of highly toxic chemicals or elevated energy inputs during the synthesis. And the executiveness of these green fabricated nanoparticles have potential application in various sectors like biomedicine, pharmacology, food science, agriculture, and environmental engineering. So this green nanotechnology method, it always follows the green chemistry principles, which are relying on preventing the use of waste, maximize atom economy, use of less hazardous chemical synthesis, safer chemicals and products, safer solvents and reaction conditions, which increase the energy efficiency with the use of renewable feedstocks and avoid chemical derivatives, and the use of catalysts, and also the design chemical and products to degrade after use, and analyze in real time to prevent pollution with minimized potential for accidents. So these are the different green chemical chemistry principles. Those are usually followed. Our lab, our research is mainly focuses on the extraction of biomolecules using the cellulose, lignin, pectin, protein, starch, chitosan. Those are extracted using the lignocellulosic biomass that are obtained from the agrobioresources, fruit waste, and the food waste. So when these extracted biomolecules come in contact with the metal salts, they form stable monodisperse composite. Those are capped with the biomolecules. And further, they are confirmed by using the various characterization techniques such as XRD, EDX, HRTM, other characterization tools to confirm the particle size, shape, and the morphology. And our lab is working to explore the applications of these nanoparticles in various sectors, mainly in case of the biomedical, that is for cosmetics, photoimaging, for the drug delivery, cancer treatment, and as an antimicrobial agent. And in case of environment, we are also studying for the sensing of the heavy metals detection. And in case of the energy sector, the enzyme immobilization using the lignin silver nanoparticles for the hydrogen production and for the microbial electrolysis cells. This is the schematic presentation of the synthesis mechanism where the metal salt with the biomolecules extracted from agro-resources on the nucleation, followed by the growth of nanocomposite, a stable and capped nanocomposite, those are formed, can be seen. The next, lignin interseeded silver nanoparticle synthesis, expanding its prophecy towards antineoplastic potency and hydrogen peroxide sensing ability. Now, why lignin? As we all know, lignin is the most abundant, renewable, and degradable biopolymer, which is commonly observed in the middle lamella of the plant cell wall. Considering its potential antioxidant and antimicrobial activity, lignin is broadly exploited for the synthesis of different biomaterials and can be studied for the drug delivery and tissue regeneration. However, it is still underutilized resource because of its certain properties such as structural complexity, variable structure based on its source, separation methods, and the low solubility. So the huge efforts have been developed to create easy, clean, efficient, sustainable, and environmental friendly procedure for nanoparticle synthesis, which follows the green chemistry principles. So lignin, as we all know, it mainly consists of chemical functional groups such as carbonyl, phenolic, aliphatic, hydroxyl, carboxyl, and thiol groups, which acts as a strong reducing and capping and stabilizing matrix in green synthesis of silver nanoparticles, making the process more cost effective and eco-friendly. Here we have determined the total carbohydrate content, phenolic, hydroxyl groups, carboxyl group, and the total phenolic content from the extracted wheat straw lignin, in which we can see the total phenolic content is found to be higher compared to the other lignin chemical composition studied. To achieve the better performance, various operational conditions were optimized, such as effect of pH, effect of temperature, effect of silver nitrate concentration, and the effect of mixing ratio of lignin and silver nitrate, in which we found the optimum temperature to be at pH 8, temperature at 50 degrees Celsius, and the 1 millimolar concentration of silver nitrate, and the mixing ratio of about 1:10 ratio was optimized. So under these optimized condition, effect of different reaction time was performed. In case of the different reaction time, it was observed that the 60 minute is the optimum incubation time to achieve the maximum lignin silver nanoparticle synthesis. As we can see, a strong absorption band at about calculated 432 nanometers indicates the synthesis of the silver nanoparticles. And this is the probable mechanism of wheat straw lignin fabricated lignin silver nanoparticle synthesis. It shows mainly the phenolic, hydroxyl, and carboxyl groups capped on the surface of the silver nanoparticles. So a stable and capped lignin silver nanoparticles were synthesized using the wheat straw lignin. Considering the stability is an important factor considering its application. So even on storage of about 3 months at 30 degrees Celsius, the lignin nanoparticles showed no particle aggregation, and also the spectrum was almost similar at 432 nanometer, but with a slight reduction, as you can see here, in peak intensity, which increases its commercial applicability. In case of the zeta potential, it showed higher negative charge, about minus 27.75 millivolts. This negative charge on the surface of the LiAGNPs were suggestive that nanoparticles, they repel each other and prevent agglomeration of nanoparticles. And due to this property, our synthesized LiAGNPs are suitable for long-term stability, which support the results of stability in the synthesized lignin silver nanoparticles. Now, this is the analytical characterization. As we can see, this is the XRD spectrum with the 4 diffraction peaks, which indicates the similar with that the standard AGNPs. And this is the FTIR of lignin and the lignin-mediated silver nanoparticles, which show the stretching of the various functional groups. Those are related to the lignin biomolecules. And this is the EDX. It shows a strong peak of silver, which indicates the presence of the silver nanoparticle. Along with it, some minor components such as carbon, oxygen, sulfur, and chlorine. Those are the biochemical components of the lignin can be confirmed through the EDX. And this is the HRTM analysis, which showed that lignin silver nanoparticles are uniformly monodispersed and are spherical shaped nanoparticles. At higher magnification, the dispersed AGNPs, they showed some fringes in black color, as you can see here, can be seen, that specifies the different functional groups or chemicals of lignin that are present on the surface of the synthesized nanoparticles. The SAD pattern exhibited the intensively polycrystalline nature of the AGNPs. Finally, the particle histogram disclosed the average particle size of about nearly 20 nanometer. The application part, if you consider, the first application of this lignin-mediated silver nanoparticle is we have studied for the photocatalytic degradation. As we know that the dye-containing wastewater, it mainly consists of toxic contaminants, that is the compounds such as surfactants, some chlorinated compounds, those are having higher COD and BOD. And because of this property, it becomes resistant to degradation. And if we discharge this untreated wastewater into the ecosystem, it affects the whole ecosystem and as well as hampers the human life. Therefore, dyeing effluent is very essential to be treated before its final discharge into the environment. There have been various physicochemical and the biological approaches that have been developed or improved to treat the dye-containing wastewater. Among these, photocatalysis is an attractive process in which the photocatalyst engrosses the light energies and consequently oxidizes the contaminant and can be a vital solution in terms of energy and environmental challenges. This process has many advantages, such as it is being inexpensive, it is rapid, and it can degrade whole contaminants with non-toxic degradable byproducts. Now here we have studied the photocatalytic degradation of reactive yellow 4G using lignin nanoparticles and optimization of the reaction limit. So to understand the effect of each factor such as only lignin nanoparticles, only H2O2, lignin-mediated nanoparticles plus H2O2 under dark condition, and finally the lignin silver nanoparticles plus H2O2 plus UV radiation were investigated. So from this graph, we can observe that the lignin silver nanoparticles along with H2O2 addition and the UV radiation were responsible for the complete degradation of this dye. And after the degradation of dye, there is no change in the color of the photocatalyst, which suggests that LiAGNP's catalyst is stable and no dye is adsorbed on the exterior surface compared to the other factors. This is the time course photocatalytic degradation of the dye developed by the LSAGNP's H2O2 and under the UV light irradiation. As we can see here, there was a decline in absorption peak intensity at 485 nanometer, and the complete elimination of peak was observed almost in 420 seconds, that is about 6 minutes, which indicates the degradation of dye by the developed photocatalytic system. For achieving the better performance, optimization of photocatalytic dosage, dye concentration on the photocatalytic degradation of dye is very important. So here we have performed the optimization condition in which in both the photocatalyst dosage and the dye concentration, the photodegradation was achieved in less time, about 360 seconds, which is lower than the other reported studies. And regarding the repeated use of developed this photocatalytic system, we know that the catalyst reusability, stability, and the lifespan. They are the important factors to develop in the photocatalytic process for long-term application. So here, our lignin-mediated nanoparticle showed a significant photocatalytic activity up to the third cycle consecutively, with complete dye degradation that was achieved, which can indicate the commercial applicability of this synthesized silver nanoparticles. Next application is the sensing application for hydrogen peroxide using lignin synthesized AGNPs. Hydrogen peroxide, that is the H2O2, it is commonly used in bleaching and as a food preservative to control the growth of microorganisms in food. However, elevated concentration of H2O2 can enhance the oxidative stress, which leads to the aging and the cancer. So here we have utilized the LiAGNPs for colorimetric sensing of H2O2, and we know that colorimetric sensing is a very easy way to find out the sensing of the H2O2. After addition of the H2O2, we could see there was a sharp reduction in absorbance of SPR at 432 nanometer, with transformation in color of the solution from dark brown to the colorless solution can be seen here, with a decrease in the absorption spectrum bands within 50 minutes was achieved, which increases our applicability of the lignin silver nanoparticles. And to develop an optical sensor, the selectivity assay, like some concomitant species, for instance, cobalt, manganese, copper, chromium, magnesium, iron, bromine, calcium, and glucose at higher concentrations were used along with the H2O2. And here in this graph, we can see a very little difference in the spectral profile related to the significant alteration when H2O2 was added can be seen. So the result indicates that high selectivity of the lignin nanoparticles for sensing H2O2 colorimetrically increases the importance and practical application. These LiAGNPs showed high sensing and selectivity ability towards the H2O2 compared with the other concomitant species. Now this figure, here we can see the probable reaction mechanism of sensing H2O2 by lignin nanoparticles. So it was supposed that after addition of H2O2, it produces highly reactive oxygen radicals which disrupt the LiAGNPs and leads to the aggregation of the nanoparticles. In the past 2 decades, we know that the human pathogens, mainly bacteria, have developed resistance against most of the antibiotics, resulting in their decreasing efficacy. So in medical science, there is an urgent need to find some benign biomaterial or bioresource for the synthesis of nanoparticles to treat microbial species with resistance against traditional antibiotics. And these nanoparticles, they exhibit high antibacterial activity due to their small size and high surface area. These silver nanoparticles usually showed synergistic effect with variety of the different antibiotics by antibacterial effect. And these mechanisms shown to be potential antimicrobial agent that can be used in various consumer products such as clothing, household, water filters, respirators, antibacterial sprays, detergent, cosmetic, and many more things. This is the probable antimicrobial activity of silver nanoparticles that takes place through entry of silver nanoparticles by disrupting the cell wall and the membrane. Then the generation of the reactive oxygen species that occurs, which further results in denaturation of DNA, proteins, and enzymes by combining with sulfur and phosphorus, and thereby promotion of intracellular ion flux finally leads to the death of the cell. Here we have checked the antibacterial activity of lignin and elevated concentration of LIAGNPs to human pathogens. The results indicate that LIAGNP showed noteworthy antibacterial potential against selected human pathogens such as Escherichia coli and the Staphylococcus aureus, with increase in the zone of inhibition, as we can see here. At different concentration, the zone of inhibition seemed to be increased. We have also determined the values of MIC and the MBC in microgram per ml and the MBC against the E. coli and Staph aureus. For E. coli, the MIC value, this one was lesser related to Staph aureus. However, MBC value for E. coli and Staph aureus, which was marginally greater compared to values of MIC in both the strains. Next application is the anti-cancer activity. As we know that cancer, it is a group of disease which causes the abnormal growth of the cell and which can spread and thus remains one of the world's most devastating diseases. In the current scenario, there are various treatment options that include surgery, radiation, chemotherapeutic drugs, and during this treatment, mostly they kill healthy cells and also produce toxicity. The rapidly developing field of various nanomaterials and nanodevices of silver nanoparticle has increased the possibility for the early diagnosis and the treatment of cancer with least adverse effects. So nanoparticles, they get attached to the metals, minerals, and drugs. This property makes a useful material to fight against the life-threatening cancer cells. This image shows the schematic diagram employed for the treatment of cancer using nanoparticles. Generally, chemotherapy is one of the known treatment process in treating cancer, and using targeted nanoparticles to deliver chemotherapeutic drugs to treat cancer provides many benefits to increase drug or gene delivery to combat against many problems associated with conventional chemotherapy. Here we have performed the in vitro cytotoxicity of synthesized LIAGNPs at various concentrations against ovarian cancer cell line SCOV3 with the standard anti-cancer drug, which is called as the doxorubicin, concentration of 25 microgram per ml. So the AGNPs were found capable to decline SCOV3 cell viability in a dose-reliant mode and at increased concentration of 250 microgram per ml, which showed about 70% of the inhibition of the cells. This is the actual cell image before and after lignin silver nanoparticle exposure, which showed dose-dependent cytotoxicity against SCOV3 cell lines. The obtained anti-cancer activity of LIAGNPs was as good as to compare commercial cytotoxic agents. So here we can also say that our lignin-mediated silver nanoparticles can be used commercially for the treatment of the cancer. Finally, based on these different applications of lignin-mediated silver nanoparticle in the area of photocatalytic degradation or the sensing of the metals or the hydrogen peroxide and for the anti-cancer activity and antibacterial activity. Finally, I would like to conclude that lignin-mediated one-step silver nanoparticle synthesis process that was developed is simple, eco-friendly, and it follows the principles of green chemistry. And the biomolecules of lignin, like hydroxyl, carboxyl, phenolic group, they act as a capping and stabilizing matrix for AGNPs. The developed LIGNPs found ideal photocatalysts and showed significant photocatalytic degradation of reactive yellow dye under optimized condition. And these lignin nanoparticles photocatalytic activity is stable and it can be repetitively used 3 times, increasing their practical applicability. And in addition, this study provides a new idea for the practice of green chemistry and a novel method for dye wastewater treatment. The LIGNPs, they also showed high sensing and selective ability towards H2O2. So developing optical sensor using such kind of nanoparticle would be easy, sustainable, and inexpensive approach to detect reactive species and toxic chemicals that increases its application in various industrial sectors. Lignin nanoparticles also showed higher antibacterial potential against human pathogens and also displayed dose-dependent cytotoxicity against the ovarian cancer cell that increases its practice in medical sciences. Finally, we believe that the synthesis of lignin-mediated silver nanoparticle is rapid, cost-effective, and eco-friendly, and thus increases their feasibility for diverse applications, including environmentally and biomedically oriented research fields. So the research
So we are running out of time, and we have to stop the presentation, and we will be moving to the next invited lecture, IL-81 by Professor Archana Tiwari from India. She will be joining online and the topic will be on impact of growth phase on biofuel production from marine diatom. Over to you, Professor. Now you can share your screen.
Good morning, everyone in Taiwan. I hope I'm audible.
Your screen, ma'am.
Yeah, is my screen being shared?
Yeah, of course. You can go ahead by PPT mode.
Yeah. So good morning, everyone. I would like to thank the organizers for giving me this opportunity to present here. Professor Ashok Pandey, special thanks to sir for this wonderful opportunity. So today's talk is going to focus on the impact of growth phase on the biofuel production from marine diatoms. I'm a professor at Amity University and my lab concentrates on diatom research. So if we talk about diatom and their application, the first question that arises that when we compare diatom to other microalgae, what are the advantages? So there's a complete holistic biorefinery which justifies the application of diatoms. When we talk about the evolution of diatoms, the cellular organization and their metabolic processes, also their ability to fix carbon dioxide is outstanding when we compare to other algal forms. Also, when we talk about the productivity, the nutrient acquisition potential, their storage ability, the potential to do photosynthesis and their unique adaptability along with their tropical flexibility is something which imparts them an added advantage compared to other classes of algae. The cellular factors that contribute towards diatom advancements include their efficient photosynthesis, CO2 selectivity, and also their metabolic pathway. I would also like to mention here that diatoms have some genes which are common to that of animal origin and some genes which are common to plant origin. So this metabolic unique metabolic setup makes them unique organisms for synthesis of multiple products. Also, if silica stress is induced, that leads to high lipid productivity, and the requirement which they have is quite less. Secondly, their amicability for mass cultivation is quite high, and when we grow them in municipal wastewater, domestic wastewater, aquaculture wastewater, we can find their efficient growth, which can be utilized for production of different bioactive compounds. So the compounds that we can derive from diatoms include nanosilica. They are the natural sources of nanosilica, hydrocarbons, sterols, carbohydrates, proteins. Also, for their application, we find their uses as biofuel in the drug delivery system, where they can be utilized as a targeted drug delivery, anti-cancer agents, and also they are the natural feed for the zooplanktons and also the different categories of fishes. Even for fish larvae, diatom is a very palatable diet. The unique bioactive compounds, they contribute towards different pharmaceuticals and the nutraceuticals which can be synthesized from diatoms. So the sustainable diatom biorefinery, it intends to enhance the biomass production by co-production of different compounds, which include the bioactive compound, also envisaging the most suitable stage. I would also like to mention here that each growth phase might not be suitable for production of all the compounds. So having a knowledge of the best phase, the best growth phase, which has productivity of that particular desired compound, can be of great interest. The tropical flexibility and the species diversity that we have in diatoms make them an outstanding candidates for production of secondary metabolites along with lipids. So the relationship between growth and lipid accumulation is something that needs to be investigated in detail so that the productivity can be enhanced, and also the identification of culture conditions which vary from species to species can be a significant advancement. So this is the whole design of the experiment that we have done. So initially diatom sampling was done. Then we went ahead with the isolation and identification of diatoms on the basis of their 18S RNA analysis and SEM analysis based on their frustule morphology. The growth was optimized, and after optimization of growth, we went ahead with their investigating the lipid and pigment production, and the best strains were screened for mass cultivation. In mass cultivation, the biomass which was generated, it was extracted for EPA, DHA, fucoxanthin, lipids, carbohydrates, silica, polyunsaturated fatty acids, protein, and in addition to it, when grown on wastewater, they were capable of phycoremediating the nitrate and the phosphate. So the work plan included the isolation of and identification. So we were not prejudiced in choosing freshwater or marine. We have initiated the study with both freshwater and marine species of diatoms, which were collected from different parts of India. And then for the initial screening, we tried to optimize the growth condition, the fucoxanthin production, EPA, DHA, different lipids and hydrocarbons. Then the best strain was selected, and for this best strain, we optimized what are the best suitable conditions for enhanced productivity, and then the lipid and pigment quantification was done, and then we went ahead with the mass cultivation. So the significance of diatoms as biofuel includes their easy cultivation, their ability to produce high yield of oil, and this oil can be directly processed into biodiesel. In addition to it, this biofuel is eco-friendly and biodegradable. There's no toxicity present, and also it is free of sulfur, which makes it a suitable candidate for biofuel. So the methodology that was adopted in the study include the initial isolation of diatoms. So for isolation, we selected the serial dilution and solid plating method. So initially in the F2 media, we cultivated the different diatom species which were obtained from diverse sites, and F2 Si media was used, and the cultivation was done at 25 degrees centigrade with 100 millimolars of light. And the single brown colonies, after subsequent serial dilution, when we obtained single brown colonies, they were handpicked and then transferred to the culture media. After the successful pure culture establishment, the identification was done on the basis of a light microscopy. Further, we went ahead with scanning electron microscopy, wherein the silica structure is a vital criteria for identifying the diatoms. So the taxonomic monographs were referred, and we also went ahead with their 18S molecular analysis. The culture conditions that we provided for this study included the F2SI media, 10% inoculum was used, and also the flask were the light and the dark duration was 12:12 hours. The biomass harvested, we in fact harvested the biomass at each phase. So it started from early exponential phase, mid exponential phase, stationary phase, and the late stationary phase. And at each step, we actually evaluated what is the concentration of different pigments along with the lipids. So the analysis that we did was the harvested biomass. Initially, we went with the gravimetric analysis, which estimated the lipids, and then the left biomass, the de-oiled biomass, pigment estimation was done. Along with that, we evaluated the primary and the secondary metabolite activity, and then the metabolites, their bioactivity was estimated in terms of their antioxidative potential and antimicrobial potential. So the biomass productivity that we obtained included a huge enhancement when we use a modified medium, that is new algae, which is fortified with nanosilica. So initial screening of diatoms for lipid production included their fluorescent microscopy, wherein we used the Nile red dye and visualized under fluorescent microscope what is the lipid productivity, and we could see that when Nile red is used as a probe, we can see the significant amount of lipids. So these are cylindrotheca, rich in lipids. The FAME profile, the GC profile, indicated the huge diversity of fatty acid along with saturated fatty acid, monounsaturated fatty acid, polyunsaturated fatty acid. So Thiocerosera, Skeletonema, Ketoceros, they showed the highest biomass concentration. Also on the basis of their gravimetric analysis, we could see that Thiocerosera could produce 52% of lipids. Also the EPA and DHA. I would like to mention here that DHA is very important for the brain development of the child, and EPA also has nutraceutical potential. So these compounds are of vital interest, and their enhanced productivity can be a great milestone in the nutraceutical industry, particularly for people who are apprehensive about consuming the animal-based product. So when we estimated the biodiesel properties, the biodiesel, any compound which is considered to be a good source of biodiesel, there are two significant parameters. One is iodine number and then the cetane number. So the higher iodine number, it represents the greater proportion of unsaturated fatty acids and polyunsaturated fatty acids. So lower IV values are desirable for standard biodiesel quality, both in terms of American standards and Indian standards. So when we estimated the cetane number, the species that we had investigated, their range, the CN number ranged from 48.76 to 48.74, which was quite promising. Also, Skeletonema had the highest iodine value, 0.712, which can be further worked on as a promising source of biodiesel. Also, the cetane number reported in the study are above the minimal CN number, which again is a very promising result. In addition to estimating the biodiesel property, we also investigated what is the EP and DHA concentration. So EP and DHA, as I've mentioned, they are great nutraceutical, and their concentration was also quite high in these marine diatoms. Again, we proceeded with the mass cultivation of the most promising microalgae. So initially, we went ahead with the indoor mass cultivation, wherein we started with 5-liter flask, and gradually we increased it to 100 liters in outdoor cultivation. So the growth curve indicates that in 10 days, we can get optimal growth of the microalgae. So these are the parameters that we analyzed in the modified media, and the media which is usually followed, the F2SI media. And we could see that the lipid productivity in case of the modified media, it was 81 mg per liter per day compared to the standard media, which was 67.9 mg per day. Also, the lipid percent dry cell weight, it was 30% dry cell weight in the modified media compared to the F2SI media. The main, the core for the study was investigating the best phase, growth phase, which could be utilized for further production, or which could be optimum for the productivity of lipids and pigments. And we could see that the yellow bars, they indicate that in case of initial exponential phase, the lipid productivity is not that high, but gradually when we proceed towards mid-exponential phase and stationary phase, the lipid productivity enhances. So when we have to harvest the cells, we need to choose the best stage which could have maximum amount of lipid. So during the early and the exponential phase, the total fatty acid was 27.8%, which was higher compared to the stationary and late stationary phase. Also in this slide, you can see the concentration of saturated fatty acid. In the exponential phase, it is 24.1, while in case of in the mid exponential phase, it decreased to 20.5, and in the stationary phase, it is 28.8, while in the late stationary phase, it is 30. When we talk about monounsaturated fatty acid, 44.5 is the value in the exponential phase, while in case of mid-exponential phase, it decreases to 39.3. While in case of stationary phase, it is 41.6, and in the late stationary phase, it further enhances to 47.7. When compared to polyunsaturated fatty acid, 31.1 is the value in case of exponential phase, while in case of mid-exponential phase, the value is the highest, 40.2. So this is the relative faint percentage and the different fatty acids that we have obtained. So in conclusion, I would like to mention that diatoms could be isolated through single pick technique, and they can be maintained efficiently in the F2SI media. Also, the FAME profiling indicates the presence of 10 diverse fatty acid in case of Thiocera phosphorgi, which includes palmitic acid, palmitoic acid, stearic acid, oleic acid, and also they constitute to approximately 86.9 to 92% total fatty acids. The saturated fatty acid and the monounsaturated fatty acid concentration, it enhanced to 43% in case of stationary phase. The polyunsaturated fatty acid exhibited contrary results. So their concentration was the experimental observations showed a reduction from exponential phase compared to the stationary phase. The polyunsaturated fatty acid, which are important role players in case of cell division, they contribute towards the polar lipid fraction, while the saturated and the monounsaturated fatty acids are the neutral lipids whose concentration is higher during the stationary phase. So the experimental results also indicate that the early and the mid exponential phase EPA productivity is high and the total fatty acid concentration was 27.8% compared to the stationary and late stationary phase wherein we get the productivity 14.7%. So the growth phase is an important factor which impacts both EPA and other functional molecule production. So investigating further on the different stages for different diatoms can give us valuable information, and enhanced productivity of biofuels can be achieved by finding out the best phase for that particular organism. So these are the publications that we have from the biodiesel work. So my laboratory works on culture media optimization, utilizing different pharmaceutical, municipal waste for growing diatoms. And also we do the indoor mass cultivation of the most promising diatoms. So that's my research team at Diatom Research Laboratory. I'm thankful to the organizers and also DBT for providing me the research funding. If you have any questions, I'm happy to answer the questions.
Okay, thank you, Professor. Thank you for your wonderful work on microalgae and in biodiesel and biorefinery application. So I have a little query. Generally, there will be the recalcitrant cell wall inhibit the production of EPA or DHA. So how you managed or you find some difference between a proper cell wall breakage protocol on EPA or DHA yield? What is your comment on that?
Thank you so much. That's an interesting question. So for breaking the cell wall, since the diatoms, they are rich in nanosilica, and breaking their cell wall and rupturing the cellular contents to have the lipid productivity, we in our laboratory, we use an ultrasonicator to disintegrate the cell wall so that we can have the enhanced extraction of lipids. So this is what we follow using ultrasonicator to lyse the cell wall.
Okay. Thank you, Professor. So
Thank you.
this will end IL 81. So the next talk will be start at 10:30, probably after 8 minutes. The talk will be IL 87 by Professor Grzegorz Piokota from Poland, and he will be talking on absorptive packed column system based on natural adsorbent for biogas upgradation to biomethane quality. So we can wait for them. Please be online, and thank you for your patience.
Organizers.
Okay, we have the pre-recorded file of Dr. Pekota from Poland, and his invited talk is on adsorptive packed column system based on natural adsorbents for biogas upgrading to biomethane quality. So we will play his pre-recorded file now. Thank you.
Organizers, at first, I would like to thank you for invitation on 10th International Conference on bioprocessing, which is held in Taiwan. And I would like to apologize that I cannot be with you physically, but here in Poland is almost 4 a.m. and outside is still dark. So I would like to, in this matter, I would like to present my presentation on adsorptive packet column system, which based on natural adsorbents for biogas upgrading to biomethane quality. My name is Grzegorz Piechota, and I'm an owner and researcher which work in the GPChem laboratory, which is responsible for research and analysis of biogas and biomethane. The order of my presentation is, first of all, I would like to give a general biomethane overview. Next, the biogas contaminants and its determination with using the chromatography methods and the different type of detections. I would like to focus on the siloxanes in biogas, which it is the main contaminant which can provide most problems if we don't remove it before forwarding biogas to biomethane upgrading installation. And I would like to tell about the sampling, type of sampling, and types of determination. The fourth is adsorptive packet column system. I would like to describe it and show advantages and disadvantages of this method for biogas cleaning. I would like to tell about the process efficiency, about how to remove siloxanes, mostly Day4 and Day5, and show you that it is possible to obtain the biomethane quality with using the adsorptive packet column system. In the end, will be the questions. The biomethane overview. We were working on the biomethane, which was provided in installation of DMT Environmental Technology. It is the company which is held in Netherlands, and he serves the technology for biomethane production and biogas upgrading using the membrane separation, the separation of CO4 and CO2 on the special membranes. The final product is a biomethane with the composition in concentration of method more than 99.8% of methane in the final product. What we should note is that in European Union now is open more than 2,500 biomethane projects which are focused on the injecting the produced biomethane into the national gas grid. In Poland, we have open almost 70 projects which are targeted on biomethane and biogas treated to biomethane quality. That can be forwarded into the national gas grid. What is important? Important is control of biogas. Control of biogas which is going to biomethane upgrading systems. So in the European Union, we have a directive RED 3, which describes the online controlling and gives the recommendation for the biogas and their concentration of each contaminants that could be forwarded for biomethane systems and finally can be injected to national gas grids. Requirements of biomethane quality injected to gas grids have several limits, mostly with the methane concentration and siloxanes, cause the initial pretreatment of biogas with removing water and another contaminants in shape of silica and non-silica contaminants are initial and are priority to achieve high effective upgrading of biogas process. The biomethane overview. So we were working on the DMT-ET technology, which you applied the special membranes which separate from biogas the CO2 and methane. The one membrane we were operated in two scales. In lab scale, when the maximum flow of our biogas was 100 cubic meter per hour and lower, and thereafter we changed the scale into the real operation one, where the flow was up to 1,500 cubic meter per hour. The efficiency of biomethane production was higher than 99.5% of methane because we multiplicated the membrane units. In this table, I would like to show you the biomethane requirements for biogas which could be forwarded to inject in national gas grids. Which parameters we can evaluate? It is the higher heating values in megajoules per normal cubic meter, VUBE index, methane number, relative density, main components, including oxygen and nitrogen, as well as H2S in the biogas stream, and mercaptans as a source of sulfur, total sulfur, CO, siloxanes. So I have on mind volatile as a contaminants, as a components of volatile metal siloxanes, ammonia, volatile organic compounds, and halogenated in shape of fluorine, chlorine, and finally we can measure the dew point. We were investigated this biomethane and biogas according to Polish standards, and we examined three different biogas which coming from landfill, agriculture biogas plants, and wastewater treatment plant. But in this presentation, I would like to focus on this last one and adsorptive packet column system. Biogas contaminants, barrier to biomethane upgrading. Yes, it is the main barrier is siloxanes and volatile organic compounds which can damage the membrane system, and the minor one is hydrogen sulfide, which is responsible for degradation of the parts of membranes. So what I can talk about the siloxane in biogas, we have two forms, two main forms. There's a form linear one, abbreviated as L2 up to L5, and the cyclic form, abbreviated as from D3, D4, and D5. Why we are focused on this components? Because the main problem is that in specific conditions which are during the biogas upgrading, the siloxanes can oxidize easily, and that leads to creation of SeO2. It is the microporous and really thin layer of the glass which can stick into the internal parts of the membrane and biogas upgrading system and damage them. Why the siloxanes are in biogas, and finally we can find it in biomethane? Because we have a polydimethyl siloxane. It is the polymer that can be used in all products or cosmetic products that we use, and during the condensation, and during the higher temperature, the cyclic form are destroyed into the linear one, and then they can easily volatilize into the volatile form, and then can appear in our biogas. And we would like to determine it in the stream of biogas and biomethane, and we apply the three ways to determine to sampling. First one is a Tedler bag that we analyze the biogas directly from the gas. Second one is the impinger with organic solvents, and third one is adsorbent tube. We use the tubes with the Tannex adsorbent and with active carbon. For us and for biomethane coming from wastewater treatment plants, the best the best approach that we obtained the results and measured results was when we used the impinger with the organic solvents. How we do it? With the biomethane was passed through the 4 impingers with the different organic solvent as a trap of the siloxanes. And in the first one, it was the material for drying of the our biomethane. Second one was an acetone. Third one was methanol, and in fourth was the N-hexane. In the final stage of this train, the gas meter was installed to measure the amount of biogas that was passing through the system. The siloxanes were analyzed with using 2 different chromatographic systems. First one is FID, flame ionization detector, and second one was with using the mass spectrometry. We use the digital system Agilent technology variant 3800, which was equipped with the 2 type of detectors. And in this table, you can see the parameters of analyze and GCMS fragmentation ions, ion 1, ion 2, and ion 3, which was the main ions for the quantitative analysis, and the retention time for qualitative analysis. Each parameters, each analysis was conducted on the column Carbovax supercapillary column, which is defined over here. What more? Why the siloxanes in biometer are danger? Because they create the SeO2. It looks like the small grain of the sun, which we forward this deposit into the SIM-ADX analysis, and we can see that in this small grain we have a Se atom concentration about 31% and O 68%. When we zoom it, we have finally SeO2. On the picture on the right side, you can see the part of the thermals where the glass deposit in form of SeO2 was formed, and we see new one and after the silica deposit. Adsorptive packet column system, the shame, a lab and real scale. The technical, so our adsorptive packet column system, which was consist of 4 separate filters, which everyone was equipped with the sampling point, was added into operated biogas installation on the wastewater treatment plant. We have 2 initial two initial columns for the biogas production: compressor, water removal system as a biogas dryer, and then we have our APCS system. The ACPS system is built for the 4 columns. Each of them contains another natural
Okay. So with this we come to the end of the last session of the day, and it was the invited talk 87, IL 87 by Dr. Pikota from Poland. And now we'll have a short tea break, and after which at 11:15 local time Taiwan we'll be proceeding towards the closing session, in which we'll be distributing the best paper awards, including the short oral presentations and the poster presentations, as well as we'll be declaring the winning for the next 11th IBA conference 2023. And we request all of the guests, delegates, as well as the audiences with us to be there with us in the closing session also. And after the closing session at 12 o'clock, we'll be having the dinner, and after which we'll be having the lunch and technical visit. Thank you so much.